All right, guys. So right now we're going to be talking about chapter 11, meiosis. And the reason why I really want to do this is because chapter 10 and chapter 11 really go hand in hand together. Um, so we will actually be talking about this a little bit tomorrow. So those of you who aren't able to make it tomorrow, this video is gonna really help you. So chapter 11. Let's talk about everything individual on the study guide. Genes versus alleles, the relationship with chromosomes, homologs or not. So this is in chapter 11. I don't know why. This actually has a lot to do with chapter 12, but I went ahead and answered it in chapter 11. Um, so genes, remember if we talked about in chapter 10, you have the DNA helix. Genes are a portion of that DNA helix that code for a particular protein. So what is an allele? Well, chromosomes going from genes, you of course have the whole DNA, which is composed of the chromosome. And I'm not gonna go through all the parts of the chromosome that was in chapter 10. But of course you have two chromosomes, or better yet, you have 23 pairs of chromosomes. But in those 23, you have one from sperm and of course one from egg. Well, those chromosomes they match up in pairs and they all code for the same thing. So you have one chromosome here. In fact, I'm going to draw something real quick. So you're going to have the way you have the chromosomes set up. Not find Joshua's phone. All right. That again. Okay, I can't do it right now. I'll type it in. So you have two chromosomes here. Say this chromosome up here at the top, this first X is for dad. We put D. First one is for mom. We'll put M. So these chromosomes here, they represent one of the two chromosomes out of the 46. The 20. So this is one of the 23 pairs. So these chromosomes, although they're not identical, they code for the same thing. All right. So whether it be eyes, eye color, have brown eyes, somebody else might have blue eyes. But when they get together, they code for the same thing. Those are alleles. When you have one or two more versions of a gene, in other words, and it's more in detail because you have different kinds. If it's for the same kind, those are I think homogeneous, and you have different kinds. Those are heterozygous. I think heterozygous and homozygous. I'm not entirely sure if I'm pronouncing that right, but yeah, those are alleles. When you have genes that are coding for the same thing, those are called alleles. All right, so that's genes versus alleles, and they really go hand in hand. There's not really a versus to it. Alleles have to do with genes. They're genes that code the same thing. Okay. All right. So that's a little bit of chapter 12 sneaking his head into chapter 11. We will talk more about this during class. I wasn't quite sure. I wasn't able to find anything on it. So if you hear the teacher say anything about ploidy level, make sure you pay attention because, like I said, I was not able to find anything on it. Okay, what is a fertilized egg? So I believe in chapter 10, if you watched the video I did, you have what's called osmolarity. No, that's not osmo. That's not right. You have I forgot what it's called. Anyway, you have what's referred to as a non-sex cell. I can't think of the name right now, but you have a sex cells and you have non-sex cells. All right. So the sex cells, of course, are the sperm and the egg. So as we talked about before, we have diploids and haploids. Sex cells are what you would call haploids, or half of a set. So if you look at a human, we have 46 chromosomes in each cell. So our sex cells are going to have 23 chromosomes. And that's important because when you have the sperm combined with the egg, they're going to form a new cell, brand new cell, and they're going to have 46 chromosomes, 23 from the father and 23 from the mother. All right. So these sex cells are also called gametes. Remember that term because it's another term for sex cell, and I'm pretty sure it's going to be on the test. These gametes come together with half a set haploid, and they're going to make a diploid. Okay, di meaning double. All right. So that's a fertilized egg is a female gamete meeting the male gamete, the sperm, and that's going to form a single. It's going to form a cell. Okay, a brand new cell that's able to undergo mitosis. That's very important because the fertilized cell, I mean the unfertilized cell, when you got just the egg and just the sperm cell, those aren't going to be doing mitosis. All right, they're not going to at once meiosis, at the end of meiosis, when you got the ending gamete, it's not going to undergo any mitosis. Okay, that's it. Until they combine, there's no mitosis. All right. So what is important? Why is it important that each gamete? Okay, we just talked about that. Okay, chromosome types, sex chromosomes versus autosomes. So we have 46 chromosomes. All right. 44 of those chromosomes are what we would call autosomes. They determine all other traits in your body, like chin shape, eye color, hair color. Okay, so that's 44 chromosomes out of the 46. The other two chromosomes are going to be those sex chromosomes, and they're going to be XX for female and XY for the male. We got that. The male's got that Y chromosome. Okay, somatic. That's what it was. So I was talking about earlier, sex cells and regular cells. So all regular cells are going to be called somatic cells. Okay, and the sex cells are the sex cells. They're the sperm and egg, and that's what we were talking about here. So everything in the somatic, everything in the body except sex cells, and the sex cells, of course, going to be those haploids. Those haploids only consisting of the 23 chromosomes in humans. I say in humans because animals are going to have other animals going to have different numbers when it comes to sex. But yes, we as humans will have 23 chromosomes in the sex cells. So what is a karyotype? So if you remember back in chapter 10, meiosis, we said we talked about karyokinesis, and I remember I said karyo is used to define chromosomes or is DNA. So karyokinesis, kinesis meaning divide, karyo meaning chromosomes, it's dividing of chromosomes. So what do you think karyotype is? Karyotype is the arrangement of the chromosomes. And this is very important when it comes to meiosis. This is the arrangement of total number of chromosomes present in the cell. Chromosomes in the same pair are called homologous. So remember we were saying how we have 23 pairs of chromosomes? Well, when I say pairs, we're referring to those homologous pairs. Okay, so 23 homologous pairs of chromosomes, and that just means we have the father, one chromosome from the father and one chromosome from the mother, and they're 23. They all look alike. They're gonna have the same length, the same features, but it's not going to have the same stuff because one's from two different people. One's from the father, one's from the mother. They're not going to be identical. They're going to be different, but they're going to have the same shape. And the reason why they need to have the same shape is because there's going to be a process later called synapsis, and they have to pretty much they intertwine together and they exchange information for meiosis. But yeah, that's karyotype. The when we talk about karyotype, we're talking about those homologous pairs. And once again, they are not identical. One is from the father, the sperm, and one is from the mother, the egg. But they code for the same things. Very important going into chapter 12 when we're talking about recessive and dominant genes and stuff like that. Mendel. So next we're going to talk about meiosis, diploids versus haploids. And we talked about this a lot now. Probably sick of hearing about it. But once again, diploids are the full set. We're talking about the 46, and the haploids are half. Well, you're going to see in the book and sometimes in the lectures, they're talking about ends and two ends and stuff. What does that mean? Well, diploids is going to be the two end. Haploids are going to be the end. And if you think about it like math, if haploids are half of the diploid, then one end at another end, and it's going to be two end, correct? So yeah, diploids, when you think of diploids, you're going to think two end. Haploids are going to be the end. So this is meiosis reduces the number of chromosomes by half. A diploid is considered two N, or having two sets of N. A diploid 2N parent cell produces 4 haploid. And that math might seem kind of odd, but we'll get more into that later. But the parent cell is going to produce 4 haploid daughter cells. In other words, the meiosis result is going to be 1 2N parent cell is going to equal N plus N plus N plus N. Not 4N, but 4 individual N daughter cells. Okay. And these daughter cells, they're not going to be identical to the parent or each other. And we will talk more about that later. So meiosis, how many rounds of cell division? So remember mitosis had just one round of cell division, and they made 2 cells. Well, meiosis is going to have 2 rounds of cell division. We have meiosis 1, the diploid 2N parent cell makes 2 haploid cells. Then we're going to have meiosis 2. Each N cell, sorry about that, each N cell is going to make, each N cell from step 1 is going to make 2 more cells, each having N. So after meiosis 2, there will be 4 cells in. There will be 4 cells in, or 4 cells containing 23 chromosomes. And that sounds very confusing, but if I can get my phone to work, I can explain that real quick. Or maybe I can use a diagram. We'll use a diagram here. My phone is acting up right now. Awesome, we got one over here. So what's happening? So one thing you have to understand in mitosis, well, before we were talking about the interphase, and remember we go through S phase. Well, meiosis the same thing. The cells go through S phase just like in mitosis. In fact, everything is the same until you actually hit the dividing phase. But understand that and during the S phase that the cells are going to be replicated. I mean, they're going to be sister chromatids, so they're going to be in this X shape here. All right, that's mitosis and meiosis. So what did I mean by what we were talking about earlier? Well, what's going to happen in meiosis? We say there's 2 rounds, and we're talking about all those ends. What do we mean by all those ends? So here we have a cell, and it's a 2N parent cell. But understand that it's gone through the process of replication. So although there are 23 sets of chromosomes, a total of 46, that all of those chromosomes have been replicated. So there's 46 pairs. So they're all going to line up in the cell. This is 1 set here. This is 2 sets here. It's going to go all the way down until you have 46 pairs. All right. So they're going all they're going to be 23 pairs rather, but it's going to be 23 pairs, total of 46. They're all going to be replicated. They're all going to be in this X shape here. And what's going to happen is they're going to split apart, and one's going to go one way, one's going to go the other way. There's two different cells. Okay, so now it looks kind of like mitosis again. And when they do this is going to be 23. That's going to be 23, and it's going to divide again. And now you're just stuck with 4 N cells. Let's show here. This is N, this is N, this is N, and this is N here. Okay, so this right here, this is going to be 2n, and when they divide, this is going to be 2n as well. Actually, is it? No, it's not, because this is 46. This is going to divide. This is 23. So this is actually n. So yeah, this is 2n. This is going to form 2n cells here and here, and those 2n cells are going to divide and form 2 more n cells for a total of 4 daughter cells that are not identical to each other or the parent. So 2 n cells divide to make two 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 n. Sorry, let me rephrase that. 1 2n cells divides to form n and n. So 2 individual n cells, and they divide to form 2 more individual n cells, which make a total of 4 individual daughter cells. So 2n is going to make that what we were talking about before, the n plus n plus n plus n. And there's a lot to really think about, but just know that in mitosis, you can go from 2n to n, and all of those cells are going to be n. All right. So mitosis talked about how many rounds. There we go. So phase 1 of meiosis, and I really I was thinking about how to do this, and if you can actually see, I actually try to draw it a little bit in the or do it a little bit in the typing of this. But in prophase, you're gonna have prophase 1. So remember, there's 2 rounds of dividing. You're gonna have you're gonna have meiosis. You have prophase 1. Metaphase 1 and telophase 1, you're going to have prophase 2, metaphase 2, telophase 2. Okay, so this is prophase 1. Prophase 1, each chromosome pairs with its homog in a pairing process called synapsis. So that mom and dad pair, they're going to combine together, all right, and they're going to kind of intertwine. And when they're intertwining, which was why I said it's important they're the same length and everything, because they have to combine together and they fit like right on top of each other, and they exchange parts. And I actually have a diagram of that right here. So what they're going to do is they're going to intertwine. It's in the process called synapsis here. And as you can see, there are 2 chromosomes of the same length. They're going to combine together, and what's going to happen is it's going to create a crossover. They're going to something called crossing over, and they're going to exchange parts. As you can see, some of mom over here got transferred over to dad's chromosome, and some of dad over here got transferred over to some of mom's chromosome. And what that it makes variability. So you have the process of synapsis and the crossing over and exchanging of DNA. And then what's going to happen is those pairs are going to line up along the plate. Now, the difference between mitosis and prophase and metaphase is mitosis and metaphase, you're going to have all 46 chromosomes line up along that line. Well, you got to think in meiosis, you first got to divide the mom and the dad ones. So what's going to happen is these chromosome pairs, I have it like here, XX, they're going to line up all the way down. X, 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 it's going to be a set lined up all the way down, one after the other. And when they're doing that, it's not going to be like father on one side, mother on one side. No, it's going to be random. It's called, I believe she called it was something random orderness or something. I can't remember. Let's see if I see it on the diagram, if they have what it was called. I do not have what it was. Yeah, here. Random independent assortment. Random independent assortment. That's what it's called. So these pairs are going to line up down the middle of the plate. And so sometimes the father's going to be over here on this side, and sometimes the mother's going to be here on this side. And sometimes, the mother's going to be here on this side. It's random. I don't think it knows how it's going to line up. But those pairs will line up and randomly, and that randomness also affects the variability of the product. if it all was the same, you know, everybody would be. I mean, what am I trying to say here? If everything happened the same, then it will increase the likability that we have kids that look alike. That when mom and dad came together on child number two, their child looked exactly the same. Okay, and you know that's not the case. When someone has children, oftentimes their children, you know, one might be high, one might be tall, one might be short. You know, one might have a different color hair. That all goes into the variability. So we were once again talking about the stages of meiosis. So once again, we have those pairs randomly lining up along the middle of the plate. And what's going to happen is in anaphase one, those pairs are going to begin to separate. We have here the X's here, and you see the distancing starting to form between the two. What's happening is the spindle is attaching to the kinetochore of those chromosomes, and they're pulling them apart. And till telophase, the homologs finish migrating to the poles at the end of the cell. So you have the distance steadily increasing in the cell until eventually cytokinesis occurs, and now you have two daughter cells. So you have one daughter cell within and the other daughter cell within, or you'll have 23 chromosomes. All right, and those chromosomes are still notice it's still an X shape. So these chromosomes are still replicated. You got that? So that's why if you were to divide them apart, you could say they were 46, but no, they're still together. So they're actually one chromosome. This is one chromosome here, and this is one chromosome here, even though they're still replicated. Until they divide apart, they're not considered two. So you have one cell here with 23 chromosomes that are replicated, and one cell here with 23 chromosomes that are replicated. So more on synapses. Early in prophase one, homologous chromosomes come together to form a synapse. Okay, we were just talking about this. Crossing over, we just talked about that as well. Okay, metaphase two. Oh, meiosis two, sorry. Meiosis two. So we just talked about meiosis one. At the end of meiosis one, we got to telophase. And if you remember correctly, telophase, we had two cells, both with 23 chromosomes, or N, two cells with N. So what happens next? Next, the phase is going to start looking similar to mitosis. And what I mean by that is in prophase, each chromosome attaches to the spindle fiber, just like mitosis. The metaphase two, the chromosomes, not the pairs, line up at the metaphase plate. So remember, the pairs have separated. They are no longer homologous pairs. We have 23 chromosomes, no pairs. Okay. So what's going to happen is it's going to look just like mitosis, and they're going to line up right along the plate, in the middle of the cell. It's going to look just like this if they were all down in a line here. So in anaphase two, the sister chromatids separate from each other. And I said sister chromatids, but they're not sister chromatids anymore. I should change that. They're non-sister chromatids. What I mean by that is the process of synapsis and crossing over has made it where those chromosomes are no longer the same. If you remember the diagram that I showed you before, I'm going to go ahead and pull it up real quick. The chromosomes are, this is the chromosome over here. This chromosome here, they're the same. Okay, this chromosome here are the same. But when you have the process of synapsis and crossing over, it results in a chromosome that is no longer the same. These are considered non-sister chromosomes. Okay, so I will go back and change that. Sorry for the error, but go back to meiosis two. So the non-sister chromatids separate from each other, and like I said, this looks just like mitosis. You have the one chromosome here, and one chromosome. As soon as they've separated now. This is now two chromosomes. Okay, so that this is the form between the two until phase two. Chromosomes arrive at opposite sides of the cell. So essentially, you have one chromosome here, the other chromosome here. They're no longer replicated. They are separated. And then, of course, you have the cytosis, which ends up, you end up in the end with an N cell. So this is an N cell, but this is an N cell as well. It's just no longer replicated. So that's how you go from a 2N cell to 4N cells that are not related. They're not identical at all. They're not identical to each other and not identical to the parent cell. But they, like I said, this process is in a way similar to mitosis. I think the main difference between meiosis 2 and meiosis and mitosis is that the sister chromatids here in anaphase, they're no longer sister chromatids. They are non-sister chromatids. And like I said, I'll go back and change that later. Okay, so mitosis versus meiosis. Oh, number of divisions. So in mitosis, the number of divisions that there's only two. One cell equals two. Those two cells are identical to each other and to the parent cell. Okay, meiosis, you end up with 4 divisions. Those four daughter cells are not identical to each other or the parent cell. And this is the same thing we're talking about here. 2 divisions here. Once again, 4 daughter cells in meiosis, 2 daughter cells in mitosis. Mitosis, they're identical. Meiosis, they're not. Number of chromosomes in daughter cells compared to parent cells. Okay, think about this one now. So remember, we said in mitosis, the goal is to make an identical two identical daughter cells. So you have 2N in mitosis, and you're going to end up with 2N in the end of mitosis. Well, meiosis, as we just said, you're going to start off with 2N, but you're going to end up with 4 daughter cells that all have N. So what does that mean? That means that in meiosis, you're going to start off with 46 chromosomes, but you're going to end up with 23 in each individual chromosome. When they divide the first time, you're going to end up with 23 chromosomes that are replicated. They're replicated. And then by the end of the second phase of mitosis, you go through prophase 2, metaphase 2, anaphase 2, and telophase 2. You're going to end up with 4 daughter cells that have undergone, that they're no longer combined, they're no longer in the replicated form, in the X form, and they all have 23 chromosomes. This is what it's saying here. Parents have 2Ns and daughters have N. 23 single chromosomes, no pairs, okay? So mitosis versus meiosis, makeup of chromosomes and daughter cells compared to parent cells. All right. We've said this numerous times. In mitosis, the daughter cells are identical to both the parent cells and each other. That's the whole purpose of mitosis, is you're going through to either grow or to repair a wound or for asexual reproduction. But when you're talking about sexual reproduction, those daughter cells are not going to be identical. We want variability. Variability is very important. So those daughter cells are not going to be identical to the parents or each other. Okay. Metaphase 1 versus anaphase 1. All right, this is more of what we were talking about. We're talking about mitosis versus meiosis, metaphase 1 and anaphase 1. So, and mitosis is not actually metaphase 1, just metaphase and anaphase, because there's only one round of dividing. Remember that. In mitosis metaphase, 46 replicated chromosomes line up at the middle of the cell, forming a plate. Oops, let me do that. In anaphase, those sister chromatids are pulled apart. So you have X here. This is just showing one chromosome of the 46. I'm not going to show all 46. I'm lazy. So that chromosome line up in the center is going to be pulled apart until it gets to the other sides of the cell, and it's going to form 2 individual cells having 46 chromosomes. Because remember, this is mitosis. But in meiosis, the 23 homolog pairs, this is metaphase 1 now, metaphase 1. So we're not talking about the one that's similar to mitosis. We're talking about the first one, the first round of dividing. So in meiosis, metaphase 1, those 23 homolog pairs. So in other words, it's going to be 23 lines. It's going to go down 23 times, and each of them are going to have two chromosomes lined up to it. These are the homolog pairs. Notice the two. 1 versus 2. 1 mitosis, 2 meiosis. So, you know, the person in the background. And so, we have the two chromosomes now lined up down the middle, further down, okay? And what's going to happen is it's going to be similar with the attachment of the spindle fibers. Those spindle fibers are going to come from the end of the cell. They're going to attach to the kintochore right here. So in the centromere, of course, you got the kintochore, right? So attached to the kintochore on each side, it's going to pull these apart in the anaphase, pull them apart until eventually they're going to go to each side of the cell, and you're going to end up with 23 chromosomes on one side, 23 chromosomes on the other. And remember, they still got one more set of dividing to do, okay? But this is 23 is not considered 46 because what? One chromosome is replicated is considered one chromosome, okay? So this is still, even though it's replicated, even when it gets to the end, it's still considered N, okay? It's not 2N, it's N. All right. So those are the difference between mitosis and meiosis. Just remember, in meiosis, metaphase 1 is going to look like this. Mitosis metaphase 1 is going to look like this. And this is just one. This is one chromosome here. This is 2 chromosomes here. Okay. I'm not going to do all 23. All right. And same thing, they're going to be pulled apart. This pulls apart that one replicated chromosome. It's going to pull apart those 2 homologous pair chromosomes. All right. So how do we allow for genetic variability? And we've been talking about that throughout this lesson. So there are 3 ways that allow for genetic variability. You have the crossing over. Remember we were talking about synapses? So synapses, those cells are going to join together and they're going to exchange parts. That's going to add to that genetic variability. So even though, say for example, the cell on the left is the one that becomes the sperm that fertilizes the egg. Well, that cell on the left, let's say that the one that goes over to this side is the father, and the one that goes over this side is the mother for that one chromosome here. Say eye color. I don't know. This is brown eye. This is blue eye. Okay. So this one goes over here, but you're also gonna have parts of it that's gonna exchange. So whether it be, I don't know, the shape of the eye or how it is or whatever, that some of that mom stuff is gonna be over here on that side. So even though the father, the dominant gene, the father gene went over here over to this side, and it's the one that fertilizes the egg. Some of the mother stuff is on that father's gene. All right, so like I said, the eye shape, you know, I don't know what it might be, but that's what that genetic variability, it adds that genetic variability, that crossing over, because they're coding for the same thing, but you're gonna have a little piece of mom on there, or if the mom came over, you're gonna have a little piece of dad with it. All right. So then you have the random independent assortment, and that was what we were just talking about. The father and the mother chromosome are going, the homologous pairs, they're going to line up in a random order. It's not going to just be the father on one side, mother on the other side. No, that's not how it works. Because if that happened, then you would just have a cell over here with just the mother chromosomes. No, we want some variability on them. That variability is a good thing because it helps, as the teacher was saying, it helps us survive. You know, if you got some people who are stronger than the other, that's that genetic variability. Okay, it has nothing to do with how strong somebody is. You know, it's about the genes. It's about the genes. Who can survive one sickness versus the other, or who can survive in one type of environment versus the other. That's all gene variability. Okay, so that random lining up in the middle of the cell. Sometimes you have father here, mother here, mother here, father here. It's all random. Okay. And then you have, of course, the random fertilization. What do you mean by random fertilization? Well, there are thousands, if not millions, of sperm cells that make its way to the egg. So when you talk about fertilization, I mean, all of those thousands and millions of sperm cells are different. None of them are the same. They all different and vary in some way or the other. So with all those different sperm cells rushing to the that egg over there. If everything is complete, we can't determine unless you do scientific where you grab a particular sperm cell. Was that vitro? No, I can't even think of the name because that's vitro fertilization is something different. It involves thank you, gene modification. So when you grab one individual sperm cell and you have all the I'm not even sure if we're at that level yet. I hope not. But when you grab that one individual sperm cell and you know, this has blue eyes, this has that, because I want my child to have this or that, and you put it into the egg. But no, when you talk about sexual reproduction through sexual means, that those sperm cells can be, you know, it can be whatever. I mean, there are so many different of them. Like I said, thousands and millions. None of them are alike. You have that's the ultimate genetic variability right there. But yes, with all those sperm cells being different, um, there is it's a completely random process. So these three make up our genetic variability: crossing over, the random independent assortment, and of course the random fertilization. It's all random. All right, nobody knows. We don't know. We can't tell ourselves what to do. As much as we was like, hey, I want my child to have blue eyes, and there's hoping, but no, it's all random. So, and then there's a lot of other traits to take effect that we're going to talk about next chapter, such as dominant gene versus recessive genes. Because just because somebody you might get the trait for blue eyes, I mean, if you got both blue eye and brown eye, one of them's going to be dominant. I think in that case, the brown eye is going to be dominant, and you know, big V versus little V, you know, big V is going to win. But yeah, we'll talk about more of that in the next chapter. How do we law for? Yeah, we just talked about that. Life cycle, 3 types. So this one, I did put some diagrams on. These are the last few slides in the chapter. But we have 3 types of life cycles. You have the diploid dominant, multicellular stage is made of diploids. And that's, of course, us. You have the haploid dominant, which are types of fungi. And that's multicellular stage is made of haploid cells. And they have alternations of generations. And these are mainly plant cells. Okay. And be sure to go that. I have also put some diagrams on that if you want to check that out. I'm not going to talk too much on it. But yeah, make sure you go. This is the diploid dominant. This, of course, is us. Then you have the haploid dominant, which is we were talking about. This is the fungi, and then of course you have the alternation of generations, and this is plants, and they go from when we say alternation of generations, they can go from diploid to haploid to back to diploid. Last question on chapter 11. Sister chromatids versus non. Okay, so this is what we were talking about. That area I have to go back and fix. In fact, I'm going to do it now after I get done with this. But when you're talking about sister chromatids, we're talking about. I'm going to use a diagram to show this. When we're talking about sister chromatids, we're talking about looking at this chromosome here. This replication has created. This happens during the S phase. Now, remember. Replication happens in S phase. It has created a sister chromatid, both having identical everything, identical genes, identical as everything's identical. Okay, nothing. This over here is identical to this. Okay, but in the meiosis, in the process of meiosis, we have what's called once again synapsis and crossover. Synapsis and crossover. This is synapsis. This is crossover. And what happens, like I said, is we create parts in the chromatin or in the chromosome that are different. And as a result, this sister chromatid is no longer a sister chromatid. They're no longer the same. And as a result, these are considered non-sister chromatids. Okay, so when we're talking about that, we're talking about dividing. They might start off as sister chromatids, but once you get to prophase 1 in meiosis and prophase 1 in mitosis, they changes the game up. Mitosis, they say sister chromatids, they split up and come to different chromosomes. But in meiosis, as soon as that prophase 1 hits in the early parts, that synapsis and that overcrossing makes those chromosomes no longer sisters. They're non-sisters because they're not the same anymore. And I believe that was everything for chapter 11. So I will see y'all on Monday. For those of y'all who I don't see, this video will hopefully help you if you watch it. You can't help you if you don't watch it. So watch this if you got some time, whenever you get the chance. Hopefully it can help you out. All right, peace out.